All right, let's talk about an organism that you're probably seeing more and more on your culture plates, Sphingobacterium multivorum. It's an environmental bug that has really started making a name for itself as a serious hospital-acquired threat. And as you'll see, it throws some pretty unique challenges our way, from the bench all the way to the bedside.
And that brings us to the big question we have to ask ourselves every single time this thing grows. Given that it lives in soil and water, is this just a random contaminant from a sloppy collection, or are we actually looking at the real culprit behind a nasty infection? That's the core dilemma we're going to unpack today.
So here's our game plan. First, we'll look at why Sphingobacterium multivorum is considered an emerging opportunist. Then we'll head to the bench to talk about identification and its challenges. After that, we'll cover the molecular methods for a definitive ID, tackle the massive headache of susceptibility testing, and finally, we'll tie it all together with clinical impact and infection control.
So let's just set the stage a little. We are talking about an organism that's perfectly happy living out in the environment, in soil, in water, but it's proven to be incredibly good at causing some really severe, often life-threatening infections once it finds its way inside a hospital.
So what exactly is Sphingobacterium multivorum? Well, it's a non-fermenting, gram-negative bacillus. The really interesting thing, and where it gets its name, is this key feature. It has high concentrations of sphingophospholippids in its cell membrane. That's actually pretty unusual for bacteria. And while for a long time we thought of it as having low virulence, it is now, without a doubt, on our radar as a significant human pathogen.
So who's most at risk here? It's a classic opportunist. We see it pop up in cancer patients, particularly those undergoing chemo, solid organ transplant recipients, people with cystic fibrosis, or patients on hemodialysis. Basically anyone with a compromised immune system. It is a master at exploiting a weakened host.
Okay, let's head over to the bench and see how this investigation usually gets started. When you get that culture plate, what are the first clues, and let's be honest, the first frustrations that you're probably going to run into?
So on the gram stain, no big surprises. It's gram-negative rods, non-motile, non-spore-forming. On blood agar, you'll see growth in about 24 to 48 hours. The colonies are pretty standard, medium-sized, circular, smooth. But the real giveaway, the key visual cue, is this distinct light yellow pigment. Now here's where the trouble starts. McConkey agar. Growth is totally variable. Sometimes it's weak, sometimes it's not there at all, and that can really throw a wrench in your initial workup.
And this is a huge headache for us in the lab. The literature is completely split on the oxidized reaction. Some papers say it's positive, others say it's negative. And that's a massive problem because the oxidized test is such a fundamental fork in the road for our ID algorithms. You get that wrong, and you might just rule out this entire group of bacteria way too early.
This is why you've got to look at the whole picture, the full biochemical profile. It's consistently catalase and urease positive and indole negative. It's an obligate aerobe. And right out of the gate, it shows off a pretty tough intrinsic resistance pattern. It's naturally resistant to aminoglycosides and polymyxins, which already starts limiting our treatment options before we've even started.
So when the bench tests are giving you mixed signals and you're just not confident, it's time to escalate. We've got to move on to molecular methods to get a definitive answer, not just a presumptive ID.
MALDI-TOF is, of course, our first stop for a quick ID. And yeah, it can identify Sphingobacterium multiform, but its success is 100% dependent on its reference library. For a less common bug like this, you might get a low confidence score or just no ID at all. That's our cue to turn to 16S RNA gene sequencing. That's the gold standard for getting a definitive species-level identification. And for the ultimate answer, whole genome sequencing not only confirms the ID beyond any doubt, but can also give us incredible insights into its virulence and resistance genes.
Okay, so we've got a solid ID. Problem solved, right? Well, not exactly. In fact, this is where the real challenge begins. How in the world do we report antimicrobial susceptibilities when we have absolutely no official guidance to interpret our results?
Like we said, this bug is tough. It's intrinsically resistant to aminoglycosides and polymyxins. But on top of that, it's really good at picking up new tricks. It frequently produces ESBLs and metallobeta-lactamases. What that means is the activity of our workhorse drugs, our beta-lactams from cephalosporins to carbapenems, is completely unpredictable unless you test it.
And this right here, this is the heart of the problem, summed up perfectly. EUCAST, the main authority for us in Europe, provides zero, zip, nada in terms of species-specific clinical breakpoints. And without those established cutoffs, we can't definitively call and isolate susceptible or resistant based on the MIC value we get.
So what does this mean for us? It means we are operating in what I call a breakpoint void. You can't just send out a report that says S or R. You have to report the raw minimum inhibitory concentration, the MIC value itself, and then you have to pick up the phone and consult with the clinical team. This really changes our role. We're not just data reporters anymore. We become essential expert interpreters, helping to guide therapy where no clear rules exist.
Okay, so let's take all this lab work and connect it back to the person it actually matters for, the patient. We have an ID, we have an MIC value. How does this translate? What kind of diseases does this bug cause, and how do we actually manage them?
And this is not a minor player. The clinical spectrum is broad and it is severe. Sphingobacterium multivorum can cause bacteremia and septic shock, meningitis, pneumonia, especially in patients who already have lung disease, and a whole range of really nasty skin and soft tissue infections, including cellulitis and even necrotizing fasciitis. Less often, it's also been found in cases of septic arthritis and peritonitis.
So when we talk about treatment, there's no magic bullet. There are no formal international guidelines, no specific Dutch guidelines to lean on. Therapy has to be completely individualized, case by case. And every single treatment decision depends entirely on the specific susceptibility results that your lab provides. You are the clinical team's only guide.
So what does tend to work? Well, if you look at a review of all the published case reports, the most consistently effective drugs seem to be the fluoroquinolones and trimethoxarim sulfamethoxazole. Beta-lactams like ceftriaxone have worked in some cases and been completely useless in others. The bottom line is you absolutely have to test every single time, no assumptions.
And you can see this in action when you look at pediatric cases. Combination therapy is super common, especially at the beginning of an infection. You can see third-gen cephalosporins are being used, but the therapy is always tailored. I mean, just look at that case of the 6-year-old with septic arthritis. That particular isolate was resistant to carbapenems and peptazo. That susceptibility report from the lab was absolutely vital for getting a good outcome.
Finally, how do we keep this thing from spreading around the hospital? It really comes down to being meticulous. It starts with strict adherence to standard precautions for everybody, with a huge emphasis on hand hygiene. For patients who are known to be colonized or infected, we add on contact precautions. That means a single room, gowns and gloves, dedicated patient equipment. And, this is critical, given how tough it is in the environment, rigorous cleaning is a must, paying special attention to sinks, faucets, and any equipment that uses water.
Just so you know, we put this entire briefing together using a comprehensive literature review from Gemini Deep Search. For anyone who wants to really dive into the data, you can view the full notebook document. It has a complete list of every single source we used to build this explainer.
So there you have it, a look at Sphingobacterium multivorum, a really challenging organism that demands our full skill set. If you found this useful, please subscribe for more explainers like this one. And more importantly, share your lab's experience with this bug in the comments below. Let's build our collective knowledge together. Thanks for your time.